The last ball, the last over of the match is always gut-wrenching. Isn't that right? Hello and welcome to Power Hours for CSIR Net June 2023. I'm your host Shekhar Suman. I hope all of you can hear me loud and clear. Welcome. आप सभी का स्वागत है. Welcome to this session, guys. I'm so glad that you are making it today. And today we are kickstarting 5 to 6 days Power Hour sessions. Okay, every day 2 hours. Our experts will come live. First, they will discuss some important questions and important concepts, and then they'll also stress upon what you should be studying, tips, tricks, techniques, and a lot of last-minute insights. And today we are kickstarting with my guru, none other than Neha ma'am. Before I bring her on the stage, I want to congratulate each one of you for not giving up, for constantly trying for all these months, and now you are in the last 6 days, right? Last 6 days, the magic is about to happen. You're about to become a JRA for a lecturership in the next 6 days. Everything is going to be decisive. What stones you pick up in these power hours are going to change the way you look at the world beyond 6th of June. So before we get started, can I have a BIO bring it on for Neha ma'am? Can you all type BIO bring it on for Neha ma'am? Then I'll bring her on the stage and we'll kickstart the power hours for CSIR. And today we are talking about Unit 1. Okay, so can I have a BIO? Welcome and good evening, everybody out there. It's such a privilege and an honor to be hosting all of you today. Bring it on, BIO for Neha ma'am. 3 cheers for Neha ma'am. She is my guru. Now let me tell you a story about Neha ma'am. She is my elder sister. Okay, that's a secret. Now, all my morning posts which you read and I write mentor, right? She's one of my mentors and she has guided me through worst of my life, okay? And the best of my life. And she has one unique ability which very less teachers have. And that is she can teach any subject by just reading it for half an hour. So she has such an amazing power of teaching ability that she just looks at this, subject which she has probably never heard of for 30 minutes, and she can teach you that in a very well manner, right? My success, all the credit goes to my guru, Neha ma'am. So let's join hands, guys. Bring it on for Neha ma'am. 3 cheers for Neha ma'am. And I'll be adding her to the screen now. Hello, ma'am, and good evening. Namaskar. How are you, ma'am?
Yeah, good evening, Shekhar sir. Am I audible?
Yes, you are perfectly audible. Thank you so much for joining for this session, ma'am, for the power hours. So let's get started without wasting time, guys. I'll pass on the controls to Neha ma'am, and she'll start the session. Ma'am, over to you. Your screen is shared. Please start with the session. Thank you so much.
Okay,
all the best.
Thank you so much. Yeah, thank you so much. So good evening, students. Very warm welcome to all of you to the power sessions organized by our company Biotechnica. And as we have always pledged, and we will continue to help you in whatever dreams or aspirations you have by teaching you the basic uh, techniques, the concepts, and everything related to help you clear any exam of life, right? Whether it is the CSIR net exam, the GATE exam, any MSc entrances, or your any Viva or any interview, right? So let's get started right now. It's time to do a quick revision for CSIR, since CSIR is around the corner. And what I have done is I have selected some very crucial concepts and the questions, and I'll be discussing all those questions and the concepts in the meanwhile to make you understand it completely and thoroughly. And in exam, if you get a question, you will be actually able to understand the question and will be able to respond to it, right? So there are a lot of biochemistry questions which can you can answer just by reading it. Only thing is you need to know the art of readability. The art of readability you have to reading ability you have to inculcate, and that is what I'm looking forward to. So let's get started. Okay. So as I've already told you, I'll be dealing with the most important concepts, and the concept starts with the enzymology, the proteins, and a little bit of your thermodynamics, right? These are the three important and crucial points which I'll be including in my today's session. And along with that, I'll be giving you the concepts, the questions, and some questions, important parts, the questions and their solutions, and then formulas and important tips and tricks at the end of the presentation. Okay, so let's get started. Fine, let's rock. Let's understand biochemistry the way it is supposed to be. Okay, so as I said, I'll be starting with enzymology, and here is my first question. Now, when you come to any enzyme any question related with the enzymes, right, you'll get a graph, you'll get some statements over there, and then you'll like panic. Okay, I'm not able to understand the graph, I'm not able to understand the statements, whether I'll be able to write that or not. So before actually reading out this question, I'll make sure that you understand the concept first, and then you yourself will be able to answer this one, right? So let's start with the concept. So the concept is starts with the enzyme kinetics. Now, under enzyme, we have what we know about enzyme. Enzyme is nothing but your biocatalyst, which actually helps in speeding up a reaction, in fastening that reaction, converting a substrate into product with by forming a small transient molecule, your enzyme substrate complex or enzyme product complex, and then later it releases the product. So this is what it does. Now during this process, we always see that there is a fastening of the process, the rate of reaction increases. Now how enzyme is increasing the rate of the reaction, right? So for that we have to actually know the catalytic power and efficiency of the enzyme. So when enzyme takes up a molecule, right, so enzyme has got an active site, enzyme takes up the molecule. Inside the active site, it actually reorients the molecule. Once it reorients the molecule, then what happens is there will be some electronic reconfiguration or configuration happens inside the molecule, and because of that the molecule itself becomes very reactive, and one substrate reacts with the another substrate to form two products, and the enzyme will release those two products. So this is how it fastens the reaction. Now in order to understand what with what ratio it fastens of the reaction, for that we have to understand the concept of enzyme kinetics. And to better start with enzyme kinetics, we have a plot which is called as direct plot. Now this is a direct plot, student. Now what is the concept of direct plot? First of all, I'll tell you that here you will see the substrate concentration. And okay, screen is not clear to Shrivani. um, I would request the backend team to actually look at it. Okay, so this plot is called as direct plot, and here what we have the substrate concentration, and here we have the initial velocity. You can see there is no reciprocals in either of the either ordinate or abscissa, right? In either of the axis, you are not seeing any of the ratios over here. That is why when a direct concentration of any object or anything is actually plotted with another direct concentration or direct number, then that plot is called as a direct plot. Okay, so this is the direct plot. And in Michaelis-Menten, for enzyme kinetics, we have one equation, that is your Michaelis Menten equation. Okay, now here what we have the substrate concentration on your x-axis and the velocity on the y-axis, and when you plot a graph you get a curve, right? You get a curve. It is very difficult to actually find out the values over here, but still this was the initial what you can say, initially this is what the scientists were able to find out, or were able to formulate, that in order to know the rate of a reaction, let's chalk out a formula, or let's find out something, and through that we will be able to know the rate of that reaction, and that is what is the Michaelis-Menten equation. So here I have the derivation of Michaelis-Menten equation. Let's go to the final formula itself, because that is not needed. The derivation part, only one thing is needed, that I will tell you later. So this is what is the formula of Michaelis-Menten equation. V naught is the initial velocity of your enzymatic reaction, and Vmax is the maximum velocity which it can attain, maximum velocity which the enzymatic reaction can attain, and this is the substrate concentration at that particular moment. This is the Km is nothing but your Michaelis-Menten constant, and which is equal to, if you follow my cursor, you can see that Km is equal to K2 plus K minus 1 divided by K1. And what is this K2, K minus 1 and K1? Let's go back to the previous slide. So it is nothing but your rate constant. Okay, so K1 is the rate constant towards the forward reaction for the formation of ES, and K2 is the rate constant for the forward reaction forming your enzyme and the product, while K minus 1 is the rate constant for the backward reaction, wherein enzyme substrate complex actually releases the substrate and the free enzyme. Okay, so K1, K2 and K minus 1, the ratio of these three will actually give you nothing but what? Km, the Michaelis constant. Okay, this is what is the theoretical value, or you can say the just the formula, this is. And what we understand by Km, I will tell you in later slides. So I think this part is clear to all of you. Quickly respond in chat, then I'll move ahead. Please, students. Okay. So now we have another plot, and that is your double reciprocal plot, or Line-Weaver-Burke plot, or Line-Weaver-Burke equation. Now already we had your Michaelis-Menten equation that was able to tell us about the velocity of the reaction. Then what is the need of this Line-Weaver-Burke plot, or you can say the double reciprocal plot. Now let's find out the name. Since Line-Weaver-Burke were the scientists which actually converted the Michaelis-Menten equation into a double reciprocal equation, right? And that is why this equation is called your Line-Weaver-Burke equation. This one I'm talking about, this equation is your Line-Weaver-Burke equation, and the graph which is plotted with it is your double reciprocal plot. Now why this is called double reciprocal plot? Because here you are plotting two reciprocals on the x and y axis. That is why this is your double reciprocal plot. Now this concept you can use in bioprocess engineering also, or anywhere. If it is mentioned double reciprocal plot, it will be like this, something like this. Now from here also we are able to get the Km, the slope gives you the Km by Vmax, and your intercept on the y-axis gives you 1 by Vmax value, and intercept on the minus x-axis gives you minus 1 by Km value. So actually if you are plotting any graph, I'm saying this is a thumb rule, guys. If you are plotting any graph for enzyme substrate reaction, or you are trying to find out a velocity, you are either plotting a double reciprocal plot or a direct plot, you are going to get all the information. What all information? You are going to get the Vmax value, you are going to get the Km value, and you are going to get the catalytic efficiency or the catalytic power of the enzyme also, right? So you will be getting all these values over there, right? Okay, is it clear? So this is your double reciprocal plot. Fine. Now I'll go back to the question now, and we'll try to find out the answer. Okay. So already I told you the concept of the direct plot and the double reciprocal plot. Now we'll read out this question, we'll try and solve it, right? So given below is the p versus time plot of an enzymatic reaction carried out by an enzyme X, right? P versus X. P is nothing but the concentration of product, and this is the time in seconds, and they have plotted a graph. Now using this graph, can you tell me something? Or you using this graph, you have to find out whether the following statement is the correct interpretation of the data or not. So here it is saying that the km and vmax of the enzyme x and x are 15 and 60 units respectively. In the first place, this is not at all a direct plot, right? This is not at all a direct plot. Direct plot is plotted against which two ordinates? Which two ordinates? What do we write in the x-axis? The substrate. What do we write in this x-axis? Yes, Yatin. No, it is not the first one. Option one is not the answer. I'll come back to that. Yes, V versus S. Right. That is what I've shown here. Please remember, it is V versus S, the substrate concentration. And what kind of graph we are getting? Here, they are giving a graph of P versus time. So it is actually not associated with any of the, you can say, the enzymatic equation graph. And by knowing that, you will know that, okay, fine, we are not going to get any information from this graph. So neither the Km nor the Vmax of the enzyme can be determined from these data. Right? So simple. Correct or not? Such a simple question. Such a simple question, right? So just by knowing small concept, just by knowing or by focusing on a very small concept, you can actually find out the answer to any enzymatic question, right? Okay. Now we will move ahead. So we learned two concepts also, the concept of your Michaelis-Menten equation, the concept of your Lineweaver-Burke plot. And along with that, I will add one more, two more actually the graphs which we see under enzyme kinetics. And one such graph is your Eddiehofsty plot. Okay, one such graph is your Eddiehofsty plot. Now, what is this Eddiehofsty plot? Using your Michaelis-Menten only or Lineweaver-Burke equation only, the double reciprocal plot, using those equations only, you will be able to find out the another relation between your V naught and V max and Km and substrate concentration. V naught, the initial velocity of the enzymatic reaction. Km is nothing but your the ratio of the rate constants. S is nothing but your substrate. Vmax is the maximum velocity attainable, which is actually a theoretical value. We can never attain the maximum velocity. Any enzymatic reaction in nature can never attain maximum velocity. It is just a theoretical value. Okay, fine. When all the enzymes will be involved in the reaction, we will be able to attain a velocity, and that is nothing but your Vmax, right? And then V naught here once again is the initial velocity of the enzyme catalyzed reaction. Okay. Now, so this is your Eddiehofsty plot. Okay. Now, what we have in the Eddiehofsty plot, you can see a reciprocal V naught by S is taken on the x-axis and V naught is taken on the y-axis, right? Okay, so we are plotting V naught by S versus V naught, and we when we plot that, we get a line like this, a slope like this. The slope will give you a value of minus Km. Okay, since it is a straight line, so y is equal to mx plus c formula we should use, right? I think some of you are aware of y is equal to mx plus c. Okay, here m is nothing but the slope of the line, right? This m is nothing but your slope of the line. So using that, you will be able to find out the slope of the line, and that will give you the km value. And vmax is nothing but the intercept on the y-axis. Vmax is the intercept on the y-axis. Now let's go back and read this question out. Now this question is saying that a plot of V by S versus V is generated for an enzyme catalyzed reaction and a straight line is obtained. So it is actually talking about Eddiehofsty equation, Eddiehofsty plot, but it's not mentioning anywhere. Now we know that, okay, it is talking about Eddiehofsty plot and what all information we can get it from that. So now you need to tell me what will be the answer to this question. So indicate the information that can be obtained from the plot. Vmax and turnover number Km can be obtained only from a plot of 1 by V versus 1 by S. Is this correct? No, this is not correct. We have just seen in Eddiehofsty equation or the plot, we can find out Km value, we can find out Vmax value, and that is a V by S versus V graph only, right? Okay, so A is not an answer. Km by V max from the slope. Km by V max is not from the slope. The slope gives you the Km value and minus Km value. So V max, Km, and the turnover can be can we find out from there? Yes. Only Km and turnover number? No. We can find out V max also. The actual answer is option 3. Very good. There is somebody named Sunshine. No, answer B is not the answer. I'll show you the graph right now. So here you can see the slope is giving you minus Km. The y-intercept is giving you Vmax value. And once you know the enzyme concentration and Vmax, you will be able to find out the turnover number of the enzyme. Okay, that is your Kcat value. Okay, so C is correct. Is this not a simple question, students? A direct question. A direct question from your enzyme kinetics. So now do not fear if there is any graph coming out of your enzyme kinetics. Okay, please do not fear. It's very simple and easy. Now, along with the information, the enzyme kinetics, there is another plot which is called as Haynes-Wolff plot, right? Haynes-Wolff plot. Now, what is this Haynes-Wolff plot? This is also another derivation of your Lineweaver-Burke equation, or you can say the double reciprocal plot only. So here, what we have they are equating, they are what do you say, deriving that a new equation from your Lineweaver-Burke equation, and that is substrate concentration by initial velocity is equal to substrate concentration into 1 by V max plus Km by V max. And when you plot a graph, you get something like this. So you are plotting S by V versus S versus S by V. Okay, always it is X versus Y, right? Whenever we talk, we always talk like that, X versus Y. So S versus S by V. And when we plot this graph, what we are getting a y-intercept, and that is nothing but equal to Km by V max. And we are getting a slope which is equal to 1 by V max. So from this Haynes-Wolff plot also, we are getting our two values, the Km and the V max value. So final line in all the plots of your Michaelis-Menten, whether it is Michaelis-Menten, whether it is Eddiehofsty, whether it is Lineweaver-Burke, whether it is Haynes-Wolff plot, all the plots, what they are going to give us give us the answer. They are going to tell you about the value of Km. They are going to tell you about the value of Vmax, and you can easily interpret the contribution of the enzyme towards the reaction. Okay, is it clear to all of you? So these were the 3 to 4 plots or the equations which I wanted to discuss along with that particular question. So I think the concept is very clear to all of you. Now, moving ahead, we have another question. And in this question, we have some more extra information they are asking, right? And here also, I'll give you all those extra information. So what it is talking about, let us read this question. So an enzyme catalyzed reaction comparing proton and deuteron transfer reactions yielded the following kinetic data. So here we have some kinetic data which is there, and that is Kcat of your hydrogen or of your proton, right, is your 70.7. Km for hydrogen or the proton is 1.03. Kcat for deuteron is your heavy water, right, an isotope of proton. So deuteron is your 10.3 and the Km for deuteron is 1.06. So you have this kinetic data with you, and what you have to do, well, you have to find out which of the following represents the turnover number for proton transfer, deuteron transfer, and kinetic isotope effect respectively. So you have to find out the kcat, that is the turnover number for proton transfer, for deuteron transfer, and for the kinetic isotope effect. Okay, the value of kinetic isotope effect. So basically, students, proton and deuteron, they are nothing but isotopes to each other. Okay, isotopes to each other. So sometimes what happens in a reaction, when you have one substrate and you put a substrate analog, what will happen to the kinetic parameter for that particular enzyme? So there will be some or the other kind of change which will be observed, and we will see what kind of change is observed, right? So we will right now learn more about your Km value. What is the concept of Km? Already we know what is the concept of Km. Then kcat value, and then the kinetic isotope effect. We'll learn these three and we'll come back to this question and we'll solve it. Okay, the same way. So let's go ahead and understand the concept first. Okay, so first of all, we will understand the concept of Km. So, what is your Km? Km is nothing but your, it is used inappropriately as an indicator of the affinity of an enzyme for its substrate. And but still we use this concept to actually find out the affinity of the enzyme. It's not an old concept. It's still being used theoretically. It is being used. So what is your Km? For any enzymatic reaction, we can write this equation: E plus S giving rise to ES complex, and then E plus P. So here we have three rate constants: K1 for this forward reaction, K2 for this forward reaction, forming ES to EP, and K minus 1 for the backward reaction. So Km is nothing but the Michaelis-Menten constant is nothing but equal to K2 plus K minus 1 divided by K1. Or you can say the rate constants for ES breakdown, the rate constants for ES breakdown divided by the rate constant for ES formation. Okay, the rate constant for ES breakdown. Okay, rate constant for ES breakdown. Okay, divided by, sorry, rate constant for ES formation. Okay, so this is what it is. Now why we need to remember this? Because as I had already shown you previously also, under enzymatic questions what we get, they will just change the statement, and you have to figure out whether they are talking about Km, they are talking about K2, whether they are talking about K minus 1, what exactly they are talking about. So you need to be aware of that. Fine. Okay, so that is why I am telling you that Km is equal to nothing but K2 plus K minus 1 divided by K1. This you all have to remember. Sometimes what happens, the K2 step becomes rate limiting. We know the concept of rate limiting, students. Anybody knows the concept of rate limiting? So rate limiting is nothing but the step which can never proceed forward until unless some extra input is given, right? Either the you can say the very strong enzyme is there for making that reaction to go proceed further, or their delta G value is not that high. So that is why the step is rate limiting. It has got a very high activation energy barrier. The slowest step, exactly, the slowest step or the highest activation energy barrier step. Now when the K2 is rate limiting, means this is reaction is actually happening very slow. Then in that case the value of K2 will be much smaller than K minus 1, wherein you can easily neglect the contribution of K2 towards the value of Km. So Km now becomes K minus 1 by K1, and that is nothing but called as dissociation constant. And in some questions Kd value is given, so you can easily use that Kd value as Km value and solve that. Okay, okay, is this part clear? You can use that Kd value, and you can easily use it as Km and solve it. Now sometimes when K2 is much bigger than k minus 1, or when this step becomes rate limiting, then in that case km value changes to k2 divided by k1. Okay, so this is a small concept about your km. Okay, now another concept which was the thing was asked in that question was something related with kcat, that is the turnover number. Okay, so here what we have, the turnover number. So for any reaction with two steps, enzyme plus substrate making enzyme substrate complex, and then enzyme plus product, and we have k1 k2 and k minus 1 adds the rate constants. So in that case v max is equal to k2 into et. This is the formula. Actually, please remember this formula. There will be a lot of other numericals also in which you can straight away use this formula. So here k2 is rate limiting, that is why we are using k2 in this equation, and multiplying it with the total enzyme concentration which is present in the reaction, and that will give you Vmax value. Now for a three-step reaction, when you have a three-step reaction in which enzyme plus substrate makes an transient structure enzyme substrate complex and another transient structure enzyme product complex, then that forms your enzyme plus product. Anusha, which concept you want me to repeat? Please mention that. So when you have the three-step reaction and when K3 is rate limiting, when K3 is rate limiting, the formula for Vmax will change to Vmax is equal to K3 into Et. So the bottom line is whichever step is rate limiting, the rate constant for that particular step can be used to calculate your Vmax value. This is another important concept, students. Okay, now K this rate limiting rate constant is your kcat. Rate constant of rate limiting step is your kcat. So in the first reaction, k2 is equal to kcat and k3 is equal to kcat. Okay, Anusha, that is related with your km value. I'll be repeating that concept once again when I'm solving numerical. So please wait for some time. Okay, I'll come back to that. Now here, as I said that in the first reaction k2 is rate limiting, so k2 is equal to kcat, and k3 is rate limiting, so k3 is equal to kcat. So v max is equal to kcat into et, right? Okay, this is the actual formula. So hence the Michaelis-Menten equation can be written as V naught is equal to K cat into Et into substrate concentration divided by Km plus S, and the K cat is nothing but your turnover number of the enzyme. For which kind of reaction? For single substrate reaction we are talking. We are not talking about the bisubstrate reaction over here. Now the constant kcat is a first order rate constant, and if it is a first order rate constant, so it will have the units of reciprocal time, that means per second. Okay, it will have the unit as per second. Okay, this will be the unit of your turnover number, the kcat. Okay, that is the unit. So in exam, if it is not they are not mentioning also the turnover number of this enzyme is this much. If the value is written and per second is written along with that value as the unit, you can easily understand, okay, they are talking about the kcat value, and you can use that to calculate the Vmax. Now that's the unit, and in normal terms we call that as turnover number, which is nothing but equivalent to the number of substrate molecules converted to product in a given unit of time on a single enzyme molecule when the enzyme is saturated with the substrate. So this is a little bit complicated, but you have to remember. So it is nothing but equivalent to the number of substrate molecules converted to product, number of substrate converted to product in a given unit of time on a single enzyme molecule when the enzyme is saturated with the substrate. So they are talking about one enzyme, one substrate, and the in a given unit of time, right? That is converted into product. So turnover number is equivalent to the number of substrate molecules converted into product, okay, per unit time. Okay, how can we know that each which step is rate limiting? The slowest step is rate limiting student. The slowest step, the highest activation energy barrier step. Okay. And usually the last step is always rate limiting. Usually, that's I should not say that is a rule, but yes, most of the time it is the last step which is rate limiting. Okay. Now let's go back to the question and try to solve this one. So here what we have. So here we have to find out the turnover number. Now they are mentioning that the turnover number, the unit of the turnover number is your per mole per second. Okay, per mole per second. That means it's actually they're not talking about the turnover number, because the turnover number, the unit has to be per second. So the value which we have in the question actually is nothing but the turnover number only. This value 70.7, 10.3 is the actual turnover number of that particular substrate. Okay, but when the same enzyme is being used, we have to use a different concept. Which concept we have to use? Now I'll tell you the concept. Which concept here we have to use? So what happens is that when the substrate concentration is much lower than km, okay, this is just a concept. Okay, this will not be given in the question, but you need to know when the substrate concentration is much lower than km, then v naught will be this v naught, the formula will become something like this, right? Because the substrate concentration is much lower. So if you add that to km, it will hardly have any impact on the km value. While when you multiply it, it will certainly have that impact. But addition will not have any impact. So you can easily remove it from the denominator. So this becomes v naught becomes k cat by km into et into s. And this is nothing but your second order rate equation. And the constant k cat by km is a second order rate constant with units per mole per second. So actually the value which they are asking is your k cat by km in the question, but they are mentioning it as turnover number, right? In the question they are asking you what is the turnover number, and the unit which is being mentioned over is per mole per second. So basically they are asking you the ratio of k cat by km. Now how k cat by km can be called as a turnover number? So let's come back to this concept over here. So the ratio k cat by km can be used to compare catalytic efficiency of different enzymes or turnover of different substrates by the same enzyme. So here in the question we have the same enzyme, but it is using two different substrates. So this ratio can be used to compare, the k cat by km ratio can be used to compare the two values, right? The two the catalytic efficiency of the two substrates, or the of the enzyme towards the two substrates. Okay, so this will give you the k cat by km value, will give you the turnover number of different substrates by the same enzyme. So here in the question you need not find out k cat value, already that is given in the question, but you have to find out k cat by km ratio. Okay, so let's go back. And so let's solve it. I've already solved this question for you. So here what we have to find out the k cat by km value. So for proton, now for proton, this is your k cat value and this is your km value. So when you do a reciprocal of these two, what you get? You get this value, k cat by km value. For deuteron, this is your k cat value and this is your km value. When you take a ratio of it, you get this value 9716. So here we have two options, in which option 1 and option 4. Here we have two options in which the values match. Only difference is your kinetic isotope effect. Now what is this kinetic isotope effect? Okay, kinetic isotope effect. So kinetic isotope effect is nothing but change in the rate of the reaction of a chemical reaction when one of the atoms in the reactants is replaced by one of the isotopes. So kinetic isotope effect is the change in the rate of the chemical reaction wherein a reactant is replaced by one of its isotope. And how it is calculated? The kinetic isotope effect is calculated by this formula: ki is equal to kl by kh. Kl is nothing but your rate constant for the light isotope divided by rate constant for the heavy isotope. Okay, rate constant for the light isotope and rate constant for the heavy isotope. So this is what we will do. The rate constant which we have calculated, okay, for both of them. So we will use both the rate constants and then we will get a ratio 7.06. So the answer will be what? Answer will be our option 4. Is this clear, students? Answer will be, what is the answer? Yes, option 4. Now, with that, I would like to give you one small extra concept, that the KIE, which we read right now in the question, the ratio of the of you can say the changes in the rate of the reaction because of the isotope changes, right? That is nothing but called as deuterium effect. That is called as deuterium effect. So in an KI experiment, an atom is replaced by its isotope and the change in rate of the reaction is observed. A very common isotope substitution is when hydrogen is replaced by deuterium. This is known as your deuterium effect and is expressed by the ratio KH by KD. Okay, KH is nothing but the rate constant for the uh, rate constant for the light isotope divided by rate constant for the uh, heavy isotope. Okay, and normal KIs for the deuterium effect are around 1 to 7 or 8. So without solving also you could have come to the conclusion that what would be the answer. So the natural value will be from 1 to 7 or 8, not beyond that. And KIE for other atoms, carbon, oxygen, nitrogen, sulfur and bromine, will be in between 1.02 to 1.10. Okay, 1.02 to 1.10. Okay, fine, students. Satvik, you can ask this question in your chat window when you are on the portal, right? Okay, please ask this question there. Fine. So we understood, and we understood the concept. Now let us go ahead and check out some more question. Okay, and this is a very simple question. Here we will learn some more concept of Km, right? So this is a very important question. So here what we have four enzyme catalyzed reaction and we have four different enzymes, EA, EB, EC and ED. So here what it is writing, A is being converted to E by enzymes EA, EB, EC and ED. The Km values of the enzymes are 10 to the power of minus 2, minus 4, 10 to the power of minus 5 and 10 to the power of minus 4 respectively. So here what we have, the Km value, okay, Km value for EA is your 10 to the power of minus 2. For EB, it is 10 to the power of minus 4. Sorry, EC, it is 10 to the power of minus 5. And for ED, it is 10 to the power of minus 4 respectively. If all the substrates and products are present at a concentration of 10 to the power of minus 4, and the enzymes have approximately the same Vmax, the rate limiting step will be, now which step will be the rate limiting step? So here everything remains the same, just the value of Km they have mentioned, and using the value of Km we can find out which step is rate limiting. Now tell me which step will be rate limiting. The rate limiting step will be the step with the highest activation energy. Highest activation energy. Okay, the rate limiting step will be the step with the highest activation energy. So anyone can tell me the answer? Which step will be the rate limiting step? Highest activation energy? more will be the value of Km. Okay, Km and affinity, they are, 1 second, hold on. So here I have written something. Let me read it out for all of you. Km is actually considered as a measure of affinity of an enzyme for its substrate, and that is where it can be used to calculate and find out which step is rate limiting step. Okay, so a high Km means a lot of substrate must be present to saturate the enzyme. Okay, a high Km means a lot of substrate must be present to saturate the enzyme, meaning the enzyme has low affinity for the substrate. On the other hand, a low Km means only a small amount of substrate is needed to saturate the enzyme, indicating a high affinity for a substrate. Okay, so low Km value means high affinity of the enzyme towards the substrate, and if the affinity of the enzyme towards the substrate is more, certainly that step will be rate limiting. So lowest Km value, which step has got the lowest Km value? Which step has got the lowest Km value? So we have four Km values: 10 to the power of minus 2, 10 to the power of minus 4, 10 to the power of minus 5, 10 to the power of minus 4. Okay, and this is for enzyme A, this is for EB, this is for EC, and this is for ED. Now ED and EB will be removed because they have got the same affinity. So either the answer will be EA or the answer will be EC. So low Km, high affinity. Low Km, high affinity. Very good. So most of you are saying the option 3. Option 3 is the answer. Acha, okay. EC is the answer. Yes. Okay, okay. Yeah, I actually missed it. Yes, it is EC is the answer. Lowest Km, highest affinity. Yes. So option 1 is the answer. Okay. Option 1 is the answer. EC is the answer. Okay. Now, yes. Okay. Now let us come to this question. This is another question, a very straightforward question, and we will solve it just like that. So here what we have, here we have an enzyme follows Michaelis-Menten kinetics. The activity of the enzyme was measured in the presence or absence of a molecule X. Okay, given below are the double reciprocal equations for the enzyme activity with or without X. So without X, this is the value, this is the formula, and with X, this is the formula. Now we have to find out what kind of molecule is this X. What kind of molecule is this X? So we have to select that from the options given below. Temperature, so this particular X is what kind of inhibitor? It is temperature independent competitive inhibitor or temperature dependent competitive inhibitor, or it is a non-competitive inhibitor, or it is an uncompetitive inhibitor. Now just by looking at the equations, how we will get to know? So for that, we have to simply remember certain concepts and some formula, and that will help us, right, with all the inhibitor related question. So let us go ahead and understand that, and then we will come back and find out what kind of inhibitor is it, right, and what is the formula, what is the change in the formula. Okay, so here we have, so let us read, go understand some concept of your enzyme inhibition. Enzyme inhibition, the act by which enzymes activity is reduced. So there are two types of inhibition: reversible and irreversible inhibition. Under reversible inhibition, we have three types: your competitive, uncompetitive, and mixed inhibition. And under mixed inhibition, we have a special category that is called as non-competitive inhibition. So we will understand all of them, and we will try and understand their graph, the formula, and accordingly we will attempt this question. Okay, fine. So what we have, so in the first one, we have your reversible inhibition. the competitive inhibition. So as the name suggests, competitive inhibitor is an inhibitor which actually competes with the substrate. So here, if you look at this diagram, if you see this diagram, you have your enzyme. So the probability of attachment of substrate to the enzyme and the probability of attachment to the of inhibitor to the enzyme is the same, right? So either es will be formed or ei will be formed. This depends upon the concentration of s and I. If the concentration of inhibitor is more, certainly ei complexes will be more, and if the concentration of substrate is more, your es complex is more. So both substrate and inhibitor, they are competing with each other for the active site of the enzyme. That is why this inhibition is your competitive inhibition. And when you plot a graph of this competitive inhibition, before that, let us find out what is the changes in the Michaelis-Menten equation. So if you look at the Michaelis-Menten equation, earlier it was V naught. What is your Michaelis-Menten equation? Always remember that. That is nothing but equal to V naught is equal to V max into S divided by Km plus S. Okay. So here we have one extra member, and that is your alpha that is attached, okay, with the km. So now what is this alpha? Alpha is nothing but a factor which actually helps us in understanding whether what is the impact of this competitive inhibitor on the value on the velocity of the enzyme. So alpha is equal to 1 plus concentration of inhibitor divided by ki. And what is this ki? Ki is nothing but the reaction equilibrium of your EI, enzyme plus inhibitor reaction. So when you have a reaction, one second. So here you have your enzyme plus inhibitor forming enzyme inhibitor complex. If you have a equation like this, for that the equilibrium constant can be written as Ki is equal to concentration of your reactant divided by concentration of product, and that value can be written over here. And when it is calculated, you get a final factor, that is your alpha. And when and this alpha is now with the your Michaelis-Menten equation, and that is the change you see. And this equation is further converted into Lineweaver-Burke equation. So this equation you have to remember, right? Okay, so 1 by V naught will be alpha km by V max into S plus 1 by V max. So with km, this alpha is attached, nothing else, right? Okay, so when you plot the graph, you will see for a competitive inhibitor, the value, as the value of alpha increases, right, as the value of alpha increases, the uh, they are when they pass on through the y-axis, they pass on through the y-axis at the same point. Okay, they make the same intercept. Okay, and nothing but that is nothing but your Vmax. So this clearly tells us that when you are adding a competitive inhibitor, there is no change in the Vmax value of the enzyme. So Vmax is not changing. Okay, Vmax is not changing. And what is changing? Your 1 by km value, minus 1 by km value is changing, right? So for a competitive inhibitor, km only changes. Km only changes. So earlier km was km, now the km changed to alpha km. Okay, this is the change. Fine. And this is how the graph is plotted. So this is the direct plot of your competitive inhibition. Okay, this is the direct plot of your competitive inhibition. Okay, fine, students. This is without inhibitor, this is with inhibitor, right? Okay, now uncompetitive inhibition. Here also there is this the same thing is there, but in this one, you the inhibitor always attaches to your ES complex. This is the difference. The inhibitor attaches to your ES complex, forming ESI complex. Okay, ESI complex it forms. So that is the only difference under uncompetitive inhibition. It is not competing with the substrate. It just attaches to the ES complex and blocks it. So there will be no production of your product. Okay, so what is the change or what is the impact which you see in the value? So the in the Michaelis-Menten equation, so V naught becomes V max into S divided by Km plus alpha prime S. So alpha prime is nothing but 1 plus I by ki prime, and ki prime is nothing but your concentration of the reactants divided by concentration of the product. So for this equation it is, right, ki prime. Okay, this equation. Now when you plot a graph, what you get parallel lines, right? You get parallel lines. So parallel lines, by that what we understand, that the V max value is changing each time. The V max value is changing 1 by V max. So when you have an uncompetitive inhibitor, it changes the V max value and it also changes the Km value. Okay, so both of it is changing, the Vmax value also and the Km value also. Okay, both of them. And this is your direct plot of your uncompetitive inhibition, the direct plot. Fine, the direct plot of your uncompetitive inhibition. Now, so there is third type of inhibition, mixed inhibition, in which we see competitive and uncompetitive, both the characters. So here you will see the changes in the equation will be your V naught is equal to V max into S divided by alpha Km plus alpha prime S. So here the inhibitor sometimes competes with the substrate. So it forms your enzyme inhibitor complex like this, and then it attaches to the substrate to form your ESI complex. So the outcome of a mixed inhibition is always a ESI complex. And and sometimes what happens here, ES attaches to I and forms ESI. So either of the ways, it will show your competitive inhibition also, it will show your no, uncompetitive inhibition also, and that is why the inhibition is called as mixed inhibition. And it is also affecting both Km and Vmax. So here you can see when the lines they cross the y-axis, they are making different what you say, the different intercepts. So different intercepts means 1 by Vmax is changing, and then similarly 1 by minus 1 by Km is also changing. Okay, so and yeah, that is your mixed inhibition. And the fourth category is your non-competitive inhibition. And non-competitive inhibition is an inhibition wherein your k minus 1 by km value doesn't change. This is a type of mixed inhibition only. This is a type of mixed inhibition wherein the minus 1 by km value, or you can say the x intercept, doesn't change. Right? This is your x intercept doesn't change. The minus x intercept doesn't change, or x prime intercept doesn't change, while 1 by Vmax changes. So this is a type of your mixed inhibition only, when you have inhibitor present and when you have no inhibitor present. Okay, and this is your graph for your mixed inhibition. Okay, now let us go back and understand. And this is your final slide. So for competitive inhibition, this is the formula: Vmax no change, Km increases. For uncompetitive, this is the formula: Vmax decreases, Km decreases. Mixed inhibition, this is the formula: Vmax decreases and Km sometimes increases and sometimes decreases. For non-competitive inhibition, your Vmax decreases and Km remains the same. Okay, so this you have to remember. This you have to remember. Okay, this is very important. Now let us go back to the question and solve it. So let us go back to the question and solve it. So what kind of inhibitor molecule is there? What changes we are seeing? We are seeing the change in the Vmax. Okay, we are seeing the change in the Vmax, and similarly we will observe the changes in Km also. That is why the inhibitor is your uncompetitive inhibitor. This is the formula. I'll show you the formula for uncompetitive inhibitor. This is the formula. Okay, this is the formula, right? 1 by V naught is equal to Km by Vmax into 1 by S plus alpha prime by Vmax. Okay, so this is the formula. Fine, students. direct and indirect plot graph, for that you have to solve, you need to have a data actually for the substrate concentration and the velocity, right? If you are trying to plot a direct plot, and if you want an indirect plot, then you need to have the reciprocals of the substrate and the velocity in order to plot that graph. Okay, and for direct plot the formula will be Michaelis Menten. For indirect plot the formula will be your Lineweaver Burke plot, Burke equation. Okay, so uncompetitive inhibitor is the answer. Simply by looking at the formula you can say. Okay, now here we have a non-competitive inhibitor, inhibition of an enzyme. Choose the plot that you would see to determine the Km. So tell me which one will be the for the non-competitive inhibitor. Which graph? This is for competitive. Okay, this is for competitive. Okay, this is for this one is for non-competitive. Sorry, this one is for uncompetitive. My mistake. Yeah, this is uncompetitive and this is your non-competitive. So this will be the answer, option 3. Yes, option C, option 3, either. Okay, so we are done with your concept of enzyme inhibition, and we are done with the types of questions which we usually get under enzyme inhibition, the direct questions we get under enzyme inhibition. Okay, so here we have another question. Now our enzymology is over, so I've just taken most important questions from the enzyme part, and that is what I have taken, and along with that I have taught you the concepts also. Now let us come to the protein part. Okay, the protein, we get another I mean more of questions from the protein part and the enzyme part, and then the metabolism part is also there, but that you have to remember, it's moreover the mugging part which you have to do with the metabolism. So now let us do the thermodynamics now here. So okay, not the thermodynamics, the protein part. So here we have a question which has already come as a part C question in a recent paper. So thermodynamics of a protein folding is depicted as a free energy funnel below. So this is the free energy funnel below, which is shown. Okay, this is the free energy funnel which is shown over here. So now given below are regions in the diagram, column X and column Y are their representation. So A, B, C and D, we have something written here. So A, and then we have some names written in the, some information is there in the column Y, and we have to match them. Okay, so under Y we have native structure, we have structure with highest entropy, we have molten globule, and we have discrete folding intermediates. Okay, now here which option is correct? Before that, let us understand the concept of your molten globule, and one more, there are two concepts, Leventhal paradox and molten globule, and this is a concept of your protein folding, students. So it's nothing is mentioned in the slide, but first of all, let me tell you the important concept of protein folding. It is observed that when a protein is produced inside, what you say, um, where exactly, what is the site of protein synthesis? The ribosome, right? So when your peptide chain is coming out of the tunnel of the ribosome, right, the first structure which it forms is your alpha helix. Okay, so there is a lot of you can say the water molecule is present. So in that particular environment, what it does is, first of all, it forms the alpha helix followed by the beta pleated sheet and then followed by something else, right? And what it does is, it forms a native conformation, and then followed by that there will be a lot of protein folding proteins. They are called as chaperones. So those chaperones will come into picture, they will help the protein to fold to their functional structure. Okay, and then the protein starts working. Now for that, there are certain models, and out of which we are going to study two models over here: the molten globule model and the Levinthal's paradox. So what Leventhal paradox says, that the um, uh, polypeptide formation or the structure formation actually is a step by step process, is a sequential process. So first of all, the alpha helix will be formed, then followed by that some beta pleated sheet will be formed, then they will combine together to form the super secondary structure, then followed by the domains, the motifs, and then followed by that tertiary structure, and then followed by that quaternary structure. So actually, if a protein starts doing that, it will take more time than usual, right? So here I have mentioned some information. Let us read that through and to understand the entire concept. So according to Leventhal, a polypeptide of 100 residues will have 99 peptide bonds, and therefore 198 different phi and psi bond angles. If each of these bond angles can be in one of the three stable conformations, the protein may misfold into a maximum of 3 to the power of 198 different conformations, which will take more than 70 years for one protein to fold, right? So do we need that kind of what do you say, do we have that flexibility of having that much of time to allow that protein to fold? No, we don't have. We do not have. So that is why this Levinthal's paradox is not taken very seriously. Now there is another concept, which is your molten globule concept. And what is this? And this says that folding is initiated by a spontaneous collapse of the polypeptide into a compact state, mediated by hydrophobic interactions among non-polar residues. The state resulting from this hydrophobic collapse may have a high content of secondary structure, but many amino acid side chains are not entirely fixed. The collapsed state is often referred to as molten globule, and that is why this particular what you say uh, uh, model is your molten globule model. So under molten globule model, what it says, that when the polypeptide is formed, it will have highest entropy, because there is no such structure to act, there is no native conformation which is formed over there. So it will have the highest entropy, and then it will do what, it will collapse. The entire of the polypeptide will collapse. It will not decide that, okay, fine, this particular structure will form alpha helix, this particular portion will form beta pleated sheet, and then we will combine and form a protein motif or a domain. No, nothing of that sort. Just the polypeptide is formed and then they collapse, and that is called as hydrophobic collapse. Why that is called as hydrophobic collapse? Because there are a lot of hydrophobic amino acids inside the protein. So all of them, they start interacting with each other, they start making hydrophobic bonds with each other, which leads to the collapse. It's just like the collapse of a building, collapse of a tower. So similarly, the collapse of the protein happens, which forms a globular structure. I mean, the theoretical globular structure, which is called as molten globule state. And further, it undergoes some kind of discrete folding to form your discrete folding intermediate, and finally forms the native structure, the first functional structure, which is formed by the protein. So this is what is asked in the question itself. So here we see that A is nothing but the protein with the highest entropy, B is the molten globule, C with the discrete folding, and D is the native protein. So D is your native protein, C is your discrete, B is your molten globule, and A is the structure with highest entropy. So what should be the answer? I think option 1 is the answer. Yes, option 1 is the answer. Okay, so this is from the direct protein folding concept. Is this part clear? And the Leventhal paradox. So now, yeah, the actual important part which I wanted to make today right now is the actual the folding. is I mean, both the paradox and the molten globule model, both of them, they are combined together, and then we say that okay, protein folds in this manner that when it comes out of the ribosome, the first primary or the first structure, secondary structure which it forms is alpha helix, and then and then not the next structure will not be beta pleated sheet or anything of that sort. It will form a molten globule to form the discrete folding intermediate, and then finally the native structure will be formed. So all these things are happening in nanoseconds, and that is how within some seconds you will have the actual functional protein. Okay, so that was your molten globule folding. Now here is another important concept and a question which I came across, and I thought I should be putting it here. So which of the following peptides, this is another protein question only. So, which of the following peptides is more likely to take up an alpha helical structure? Okay, to take up an alpha helical structure. So, which one? There is two peptide, and out of which will take up an alpha helical structure. Now nothing in no information is given except of the delta G value, and then some single letter or your amino acid is written LKAEN, and it will be very difficult to actually depict which amino acid sequence is this one and this one. It will take too much of time. So the easiest way is what? The easiest way is to actually look at this value, the delta G value. The delta G value of the first one is minus 13, and the delta G value of the second one is plus 41. So which will show you the what you say, the exothermic or the irreversible reaction or thermodynamically possible reaction? The first one, right? So the first one is actually thermodynamically more possible than the second one. So as I had already told you, in the ribosome, the first the one once the polypeptide comes out of the ribosome tunnel, the first structure which it takes up is the alpha helical structure, right? And that is the most thermodynamically possible structure. So here we have, since the first one has a delta G of minus 13 kilojoule per mole, and the second one has a delta G of plus 41, so the first one will have more chances of forming an alpha helical than the second one. Yes, spontaneous, Abhijit, yes. Okay, so this is very important concept, that if thermodynamically possible structure is your alpha helix. Okay, now the next question: which of the following statements on protein conformation is not true? So here comes the Ramachandran plot, the concept of Ramachandran plot. So before dealing with this question, let's understand the Ramachandran plot. So what is your Ramachandran plot? Levinthal's paradox. Levinthal's paradox actually says that the protein, when it is formed, will take up alpha helix structure first, then followed by that beta pleated sheet structure. The secondary structures, the protein polypeptide will form, then it will form the super secondary structures, then it will form the tertiary structure, and then followed by that it will form the quaternary structure. So when a protein is actually undergoing say folding, when it forms the alpha helix structure, there will be phi and psi angle. So there will be a lot of permutations and combination in order to get the actual structure of the actual structure with least energy, right? So it is very difficult to actually formulate that, or Levinthal's paradox, to use that Levinthal's paradox, because it is saying that the protein folding is a step by step process, and if it is happening like that, it will take some 70-80 years to actually fold one protein, which is not possible, right? A protein folds in a nanosecond, right? So in order to have that, what we the actual protein folding concept says that initially the first structure which is formed is your alpha helix, but after that, once the alpha helixes are formed, then the protein it forms a molten globule first, then followed by discrete native structure, and then followed by the native structure. Okay, so this is what Abhijit it is, right? I suppose you understood that. Yes, I think it is very correct. That gives us the actual confirmation of your the possibility, the possible confirmation of the protein in nature, right? In not in nature, but the actual possible confirmation which a combination of peptides can take. That is what the Ramachandran plot depicts. So here we have two such questions, and we will just first of all read the Ramachandran plot. So Ramachandran plot is a plot which actually tells us about the phi and psi angles, right? Are you aware of the phi and psi angle, students? I think I missed that. Okay, so when a polypeptide is formed, right, you have C. Uh, yes. Okay, like this, some structure like this. So basically the peptides are very much planar, right? The peptides are planar. Why? Because they have this double bond character, and because of which the rotation across the peptide is very restricted, right? So Ramachandran plot actually tells you about that phi and psi angle, the angle which nc alpha and cc alpha, n c alpha and c sorry cc alpha can take. One second. This C alpha. Okay, C alpha will take. So NC alpha will make what? Phi bond. Okay, and this one CC alpha will make what? Psi bond. So the actual phi and psi angles which a peptide can take up in your nature or in your 3D space, that will allow a particular conformation of a protein. That is what your Ramchandran plot tells us about. Okay, so the Ramchandran plot is the 2D plot of the phi and psi torsion angles of the protein backbone. It provides a simple view of the conformation of the protein. Conformation of the protein means the three-dimensional structure of the protein. That is what it tells us. Now, phi and psi angles clusters into distinct regions in the Ramachandran plot, where each region corresponds to a particular secondary structure. Right. So here in this one, in this Ramachandran plot, you can see here it is alpha is written. That means alpha helix. Here it is beta, then beta pleated sheet. Here polypropylene structure is seen, and then here what you have, the left-handed alpha helix is observed, right? So phi and psi. This is your phi angle. This is your psi angle. Okay, so these are the allowed regions in which you see these structures. Okay, so there are basically four types of Ramachandran plot depending on the stereochemistry of the amino acid. One is your generic Ramachandran plot, which we always discuss. Another one is your glycine Ramachandran plot, the proline Ramachandran plot, and the pre-proline Ramachandran plot. Okay, I'll show you all of them first, and then we will discuss some more information, right? So this is your Ramachandran plot for glycine. You can see the presence of glycine everywhere. You can see the dots, right? The small dots. Can you see all the dots? So these are nothing but the allowed conformation of the glycine. And where you see the maximum concentration of the dots, that is the maximum allowed conformation of the glycine. So it will be found in your right-handed alpha, your left-handed alpha, your beta pleated sheet structure. It will be found everywhere, right? Why? Because if you see, look at the structure of glycine. It is the simplest of the amino acid, and it doesn't have a bulky R. In place of bulky R, it has got a hydrogen. So it is very simple or flexible amino acid, and it can be found in any conformation: alpha, beta, right-handed, left-handed, everywhere. Okay, now what about proline? As I said, that there are only four types of Ramachandran plot which we are going to study. One is generic, another one is glycine, proline, and proproline. So this is your proline structure. Now proline Ramachandran plot. So this is your proline. If you look at the structure of proline, you can see it has got a restricted movement, right? Because of this pentagon structure. right? This pentagon structure makes the proline very rigid, and it doesn't allow that particular amino acid to be a part of any alpha helix. So once an alpha helix is being formed and a proline comes into picture, so from that place only the alpha helix gets distorted and it doesn't continue being an alpha helix. So some other structure, a loop or a loop will be formed, or something of that sort will be formed, or beta plated sheet structure will be formed, but the alpha helix structure will be disrupted once the proline comes into picture. So you can see the allowed regions of proline is the right handed alpha and the polypropylene region, right? These are the only two regions wherein you will see proline in maximum concentration, and here very less, and here also not that much, right? So these are the only two allowed regions of proline. Now there is another one, that is your pre-proline. Pre-proline is nothing but your residue preceding a proline. So when proline is being made, an intermediate which is formed before proline is your pre-proline. Okay, so you can see the orientation, or the pre-proline is not that restricted. You can find pre-proline structure in this particular area under 0 to plus 180 degree of psi, and 0 to minus 180 degree of phi, right? In this area you can see more of pre-proline, and in the very small in this one, and here also in alpha helix also, right? The pre-proline. So yes, so this is all about your the small information about the Ramachandran plot. Okay, the small information. Now, let us read the question and find out whether whatever we have read till now, can we use that information to solve this question? So, which of the following statements on protein conformation is not true? That it is asking which one is not true. Dihedral angles of side chains in amino acids are depicted in Ramachandran plot. No, not at all. Dihedral angles of the side chains in your what you have in your Ramachandran plot, they talk about CC alpha and NC alpha, right? The R, wherever the R is there, right? Wherever the R is there, it is out of you can say context. Only this part is involved, the CC alpha and NC alpha, right? Nothing else is involved. So dihedral angles of the side chains are not at all involved, right, in the protein conformation, the 3D protein conformation of the Ramachandran plot. Now, the infrared spectroscopy can be used to deduce hydrogen bonding in peptides. Okay, so can we use infrared spectroscopy? Yes, it is used. The three-dimensional structure of protein composed of 100 amino acids can be obtained by NMR. Yes, NMR is always used to find out the 3D structure of your protein, and then globular proteins have alpha helical and beta sheet components. Yes, can you give me any example of globular protein which has got alpha helix and beta pleated sheet also? And it is very popular. I'll give you a hint. It has what it has. It carries oxygen in the blood. So that's the biggest hint which I can give you right now. So a globular protein which has got alpha helical and beta pleated sheet structure. Please quickly respond in chat. Hemoglobin. Yes. It was easy to guess. Yes, hemoglobin. Hemoglobin is a globular protein which have alpha and beta pleated sheet structure. Okay, so 4 is also correct. So which is not correct? Dihedral angles of side chains in amino acids are depicted in the Ramachandran plot. No, it is not. It is not at all depicted. Yes. So now the next question, let us come to the next question. So what we have given below are statements related to protein structure. So what and we have to read through all these statements, and then we have to find out all correct combination, right? Which combination is correct. So the dihedral angles of an amino acid X in acetyl X methyl amide. So here we have a tetrapeptide acetyl X methyl amide in the Ramachandran plot occur in very small but equal areas in left and right quadrants. It can be concluded that X is not one of the amino acids. Probably this is a pre-proline. Okay, this could be pre-proline, but still let us read some more statements. So the dihedral angles of 20 residue peptides are represented in the Ramachandran plot. It is possible to conclude that the peptide doesn't have a proline. When the dihedral angles of the 20 residue peptide is represented already, proline is also represented. We cannot say that the peptide doesn't have a proline, right? Because proline is also represented in the Ramachandran plot. Yes, it has got very small place, right? Because of the restricted structure, it cannot produce what you can say various types of phi and psi angles, because it cannot rotate and move and form a different conformation when it is present in a protein, but still it is a part of a Ramachandran plot. Okay, so two proteins can have similar fold even when they do not share significant similarity in the primary structure. Yes, they can, because it depends upon the motif and the domain which they form, right? So they can have a similar fold, they can have similar motif and domain also, right? So they can have on denaturation of a protein by urea, the interaction that would be disrupted are ionic bonds and van der Waals interaction, but not disulfide bonds. That's correct. Denaturation of a protein by a urea disrupts only ionic bonds and the van der Waals. It doesn't disrupt disulfide bonds. What we need to disrupt the disulfide bonds if we are denaturing a protein, or for a protein denaturation? Not hemoglobin. Here, hemoglobin is not the answer. Beta mercaptoethanol. Okay, so all correct is A, C, and D. Okay, all correct is your A, C, and D. I've already explained to you. Yes, yes. Okay, fine. So this was something related with your Ramachandran plot, and along with that, I gave you the concept of Ramachandran plot also. Okay, now some more questions under protein. What we have? So following are statements on beta turns of protein. Beta turns. So protein has got loops, protein has got turns, right? So a loop is found when an alpha helix is disrupted, a proline comes into picture. So the alpha helix gets disrupted at that point, a loop will be formed, right? It is not a, it is a part of your secondary structure only, but it cannot be called as a secondary structure because it's not a fixed structure. The loop is not at all a fixed structure. But yes, in a polypeptide, it is fixed because the position is fixed. So it is a fixed structure. Now, let us read the statements and find out about the beta turns. So, following are the statements on the beta turns. All the 20 coded amino acids have equal propensity to form beta turns. All of them will form beta turns? It's not so, right? All of them with the same intensity they are going to form beta turns? No, not at all. Proline cannot occur in beta turns. Proline always occurs in beta turns, right? Proline can always be found in turns and loops. Whatever turns and loops you read, always a proline will be there. So B is incorrect. So when B is incorrect, so this is incorrect. Okay, now C. Proline and glycine sequence strongly favors beta turns. Yes, C is correct. C is completely correct. In aspargine glycine beta turns, aspargine can have positive phi and psi values. Certainly, it can have positive phi and psi values. Okay, fine. So the actual answer will be your C and D, right? Because all the 20 amino acids will not have equal intensity to form beta turns, and proline cannot occur in beta turns. Okay, right? Fine. So C and D are the answer. Now I came across another question. It is associated with the stability, and I thought I should discuss this one over here. And here, what we have, a researcher has developed a program to evaluate the stability of a protein by substituting each amino acid at a time with the other 19 amino acids. For a protein, a researcher has observed the following changes in stability upon substituting amino acids in loops, helices, sheets, protein core, and on the protein surface. So here he has plotted a graph, and looking at the graph, we have to find out that substitution in loops are more tolerant. I mean, which of these statements are correct? So substitutions in loops are more tolerant, substitutions in sheets are more tolerant, substitution in core is less tolerant, substitution in helices are less tolerant, and surface is more tolerant. Actually, if you look at the three structures, the loop, and the surface, two structures. Here you can see the stability is increasing when you are having a substitution. So loops and surface, they are more tolerant, and that is why we see more stability in them. Whether you do any substitution, whatever you do, there is more stability, so it is more tolerant, right? From the graph we can make out. Alpha helix, if you look at, so alpha helix has got a decrease in stability also, as well as increase in stability. Well, if you change or if you substitute any amino acid over there, right? If you substitute a specific amino acid, the stability is increasing. If you substitute another amino acid, it the what you say, the what is that, the uh, stability is decreasing. So here loops and surfaces are more tolerant, and then here sheets and core, they are less tolerant. So sheets are more tolerant, this is correct. Incorrect. Helixes are less tolerant, this is incorrect. Core is less tolerant. So A, C and E are correct. So let's find out wherein we have A, C and E. So we have in option A and C, and in other we have B and E. Since B is incorrect, so we'll not consider this one, we'll consider option 1 as the answer. Is that clear? Option 1 as the answer. Yes. Okay. So now the next set of question which we get will be from the hemoglobin. Okay. So first of all, I'll teach you a little bit about the hemoglobin, the sigmoidal curve, and then we will come back to this question. Okay. So let us read the hemoglobin concept. Okay, fine. Why I'm teaching you this? Because as I said that in your part C question or part B question, if a globin is written, so certainly if you know about hemoglobin, you can straight away use the idea. And whatever information I'm giving about hemoglobin, same you can use for myoglobin and cytochrome, right? The same information. Almost everything is equal except for some kind of dissimilarity. So hemoglobin, as the name suggests, it has got two parts, heme and globin. So here we have your heme part, okay, and this is your globin part, okay. The brown colored and the pink colored structures are nothing but the globin part. That's the protein part. So hemoglobin is made up of protein, and this is your protein, and this is your non-protein part. Non-protein part is also called as prosthetic group. We have already discussed that, right, under enzymes. So the heme has got, now let us talk about the heme first. Now heme has got an iron in Fe2 plus conformation, okay, Fe2 plus. So the coordination number over here is 2 plus, it's not Fe3 plus, okay. It is not, one second, it is not Fe3 plus. Please remember that. I will tell you why it is not Fe3 plus in the later part of the slide. So it is Fe2 plus conformation, and it can make 6 coordination bonds, right? 4 with the nitrogen and 2 here and there, right? One with the oxygen and another one with the globin, right? The protein, okay, another one with the protein. Okay, this is how the protoporphyrin ring is sitting on the globin. So in hemoglobin we have 4 subunits, 2 alpha chain and 2 beta chain, right? The brown ones are your beta chains and the pink ones are your alpha chains. And with each of the chain, what do you have? We have one protoporphyrin. So there are 4 polypeptides, and each polypeptide has got one protoporphyrin. So there will be 4 protoporphyrin, and one iron will actually accept one oxygen at one time. So there will be 4 oxygen molecule which can be carried at one time with with the help of your hemoglobin. Okay. Now I think I have already discussed this part. So let's quickly go to the go to the next slide. Yes. So what is this? Yeah, under hemoglobin, what we have, we see there are two states which are formed: T state and R state. R state is your relaxed state, which is attached with oxygen, relaxed. So when hemoglobin is attached with oxygen, which is called as relaxed state, and when it is not attached with oxygen, it is tensed. Okay, so relaxed oxygen, tensed no oxygen. Okay, now, so here we have your T state and R state. T state is the tense state without oxygen, and R state is with oxygen. So under R state, you can see the positively charged side chains are coming out. Okay, the positively charged side chains are coming out, and under T state, you can see the negatively charged side chains. So when the protein, the hemoglobin, takes a three-dimensional form, you will see that when it is in the R state, when it is with the oxygen attached, it will have positively charged side chains coming out of it. And then when it is under T state, when it doesn't have oxygen, then there will be negatively charged side chains coming out of it. This is an important information. Now, yes, now with the hemoglobin, we have one very important information, and that is there is a sigmoidal curve which is observed when under your when hemoglobin attaches with the oxygen, and which is called as oxygen hemoglobin dissociation curve. Since this is S-shaped curve, right? So this is called as sigmoidal curve, right? Now we see that this sigmoidal curve, sometimes it shifts towards right. Okay, it shifts towards right. So sometimes what happens is it will make like this, something like this, and sometimes it shifts towards left, something like this, right? So when it shifts towards right, when it is seen when there is decreased affinity for oxygen, when there is increase in partial pressure of your carbon dioxide, when there is your less pH, when there is more of bisphosphoglycerate. Which bisphosphoglycerate? 2,3 bisphosphoglycerate. Earlier it was called as diphosphoglycerate, now it is called bisphosphoglycerate. So 2,3 bisphosphoglycerate, and when there is high temperature, so you will see that this sigmoidal curve actually shifts toward right, and that is called as right shift. And sometimes you will see this sigmoidal curve is shifting towards left. When it shifts towards left, when there is less of partial pressure of carbon dioxide, when there is uh, uh, less of H plus, that means more pH, when there is less of 2,3 bisphosphoglycerate, when the temperature is low, and in your fetal hemoglobin, you will always see a shift towards the left hand side. Okay, so this is always observed under tissues. Okay, under tissues, this is usually seen in tissues, and this is seen in the lungs. Okay, and in the lungs. Okay, this is seen in the lungs, and this right shift is usually seen in tissues. So this is just a small information. Now let us go back and solve our question, the two questions which we have. Yeah, this one. Okay, now it is talking about the dissociation curve. So oxygen hemoglobin dissociation curve illustrates the relationship between PO2 in blood and the number of O2 molecules bound to hemoglobin. The S shape of the curve has been explained in the following proposed statement. So here there are some statements, and we have to find out the incorrect statements. So the quaternary structure of hemoglobin determines its affinity of towards O2. The quaternary structure of hemoglobin determines its affinity towards O2. We'll just keep this statement. We'll find out whether this is right or not later. Now, in the second one, in deoxyhemoglobin, the globin units are tightly bound to a T configuration. We have not seen that, right? We have not seen that, but still we'll just keep it. Now the C, what C is saying, the interactions between globin subunits are altered when O2 binds with deoxyhemoglobin. Yes, certainly, when the O2 binds with deoxyhemoglobin, we have already seen the state gets converted into relaxed state, and under relaxed state, which type of amino acids will be protruding outside, the positively charged or the negatively charged? Which one? I've already shown you that. Here, the positively charged, right? Under relaxed state, when oxygen is attached, right, the positively charged amino acids will be protruding outside. So yes, there will be interaction between the globin subunits are altered when O2 binds to deoxyhemoglobin. Yes. Now the affinity of O2 in T configuration of hemoglobin is increased. This is completely wrong, right? This is completely wrong. The affinity of O2 in T configuration is increased. How it is increasing? How it is increasing? Do you know the attachment of first oxygen in T configuration is much higher, much difficult than the next oxygen molecule attachment, right? So basically, what happens is the first oxygen when it attaches to the hemoglobin is in T state, right? And then T state starts converting into R state, right? The T state starts converting into R state. So once first oxygen say attaches to the hemoglobin, then there will be some alteration in the structure. Then immediately the second oxygen will be attached. So the affinity will be little bit increase, little bit increase. Then after that, the third oxygen molecule will attach. So the affinity will become once more again it will increase, and for the fourth one it will be very high, right? So the attachment of fourth oxygen molecule to the hemoglobin will be very fast as compared to the attachment of the first oxygen molecule. Okay, so this is completely incorrect. In the relaxed configuration of hemoglobin, the affinity of O2 is reduced. No, not at all. So the D and E statements are completely incorrect. Now let us justify whether A, B, and C are correct or not. Okay, so the quaternary structure of hemoglobin determines its affinity of O2, affinity to O2. Yes, if the quaternary structure is not formed, if it is a primary structure or say it is a tertiary structure, certainly the affinity of O2 will be much lesser, because the attachment of one oxygen actually coordinates the attachment of another oxygen, right? As I said, the first oxygen attachment will take more time, then the second oxygen attachment, then the third oxygen attachment, and once the three oxygen is attached, the fourth one attaches immediately. So the affinity increases, right? So the quaternary structure of hemoglobin determines its affinity to O2. Completely agree with that. In deoxyhemoglobin, the globin units are tightly bound in a T configuration. Yes, they are tightly bound. The interaction between globin subunits are altered when O2 binds with deoxyhemoglobin. Completely agree with that. So that is where the incorrect statements are only D and E. Yes, that property is cooperativity. Exactly. I think I said coordination. It's not coordination, it is cooperativity. I have already mentioned that. Okay, so hemoglobin is an allosteric protein. What is the concept of allosteric protein? Anybody? Allosteric protein. A protein which never stops, right? It never stops. Its activity can be decreased and increased, but it never stops, right? Same is with the enzyme, students. Allosteric enzyme, the enzyme will never stop. It's just that sometimes its speed will be much higher than the rest of the times, right? But it will continue with its work or with its activity. That is the meaning of allosteric in nature. Yes, that is cooperativity changes in the structure with substrate addition. Exactly, I completely agree with you, Yatin. So the interaction between heme and oxygen is termed homotropic modulator, because here the ligand and the activator is the same. The ligand is also the oxygen ligand, just like for enzyme we say the substrate. Similarly for proteins, we say that that is a ligand. So ligand as well as the activator is the same. That is why the hemoglobin is a homotropic protein. Okay, so this is all about your hemoglobin, and I've summed it up in your oxygen, the oxygen dissociation curve also. This you should always remember. This will help you in solving almost all questions which will be part of your unit 7 as well, right? Okay. Now we have another question. It is once again related with your hemoglobin only. Let us find out whether we can answer this one or not. CO2 dissociation curves or of oxygenated and deoxygenated blood are given along with dissolved CO2 below. So you have deoxygenated blood, you have oxygenated blood, and you have dissolved CO2. The graph is given. Following are the statements deduced from the curves which above and or based on the knowledge about CO2 transport, which may or may not be correct. Okay, so we have to find out whether which all are correct or incorrect. The deoxygenated hemoglobin has greater affinity for CO than oxygenated hemoglobin. Yes, this is very true. The deoxygenated hemoglobin has always got higher affinity towards carbon dioxide. It should be CO2, it should not be CO. I think by mistake I wrote CO, so it should be CO2, right? So it always has a high affinity towards CO or CO2. And anybody can tell me what is the attachment site of CO2 to the hemoglobin? Where exactly the CO2 attaches to the hemoglobin? Anyone? Where globine chain, that's correct. The amino terminal region of the globine chain. Okay, so CO2 always attaches to the amino terminal region of the globine chain, and place where oxygen attaches is the iron. The iron which is centrally placed at in the protoporphyrin ring, right? So the oxygen attaches to the iron. Okay, so CO2, the attachment site of CO2 is the amino terminal end of your globine chain. Okay, amino terminal end of the globine chain. Now, the deoxygenated hemoglobin doesn't bind with free H plus ions released during the formation of HCO3 minus from CO2. I'm not aware of this information. I'll just check it out. 1 minute. The deoxygenated hemoglobin doesn't bind with free H plus ions during the formation of HCO3 from CO2 minus. I suppose yes, it doesn't bind. This option has to be correct, but still let us check some more statements. The hemoglobin saturation with O2 has no effect on CO2 dissociation curve. It will always have. So this statement is wrong. Okay, it will always have. O2 and CO2 bind to hemoglobin at different sites. This is correct. So what we have A, B, and D. So we have yes, A and D. So option 4 will be the answer. Okay, option 4 will be the answer. Fine, students. Now, here, if you look at the question, the information which is given is completely different than what they are asking in the option, right? The option is more associated with your dissociation curve and the ability with which hemoglobin transfers oxygen to the tissues, or it takes up carbon dioxide from the tissues, right? So this is how a part C question is framed sometimes. The question is depicting something, asking something else. So please be aware of that. You increase the best reading ability of the question paper. Okay. So now let us go to the next set of question. Okay, so here we have fluorescence emission spectra of three proteins. Now this is based on the direct information, students. Here you have to mug up, but still I will make it clear to you. So let us read out the question first. So we have a graph wherein fluorescence intensity is on the y-axis and wavelength is on the x-axis. Wavelength is nothing but your lambda. Okay, lambda is your wavelength. And here we have three proteins X, Y, and Z. So above figure shows the fluorescence emission spectra of three different proteins, protein X, protein Y, and protein Z, excited at 280 nanometer. So all of them are excited at 280 nanometer. So most probably they will have tyrosine, tryptophan, and all those, right? Now, which one of the following statement gives the correct interpretation? Okay, let us find out which one is giving the correct interpretation. Proteins Y and Z have tryptophan, while protein X has only phenylalanine. Okay. Protein X has only tyrosine and protein Y has tryptophan on the surface, while protein Z has tryptophan buried inside. Protein X has tryptophan buried inside, while proteins Y and Z have tryptophan on the surface. Protein X has only tyrosine and protein Y has tryptophan buried, and protein Z has tryptophan on the surface. Okay, so here we have statement number 2 and statement number 4, which are just opposite to each other. So most probably in this kind of question, I mean, that's the trick. The two options which actually are very similar could be one of the answer. So let us first understand the graph, and then we will come back. So here we have a graph of tyrosine and tryptophan. Okay, so tyrosine, both of them, they are excited at 280 nanometer only. Okay, and they show the wavelength till 400, right? So generally the peak we see in between 300 to 400. Okay, yes, emission, and very very good, Yatin. Emission at 305 and free tryptophan at 350 nanometer, very good. So tryptophan in solution displays an excitation maximum near 280 nanometer and emits near 350 nanometer. Okay, so here what we are seeing is tryptophan in solution displays an excitation maximum near 280 nanometers. So basically, tryptophan excites at 280 nanometer and emits near 350 nanometers. That means 300 to 400 nanometer. So probably protein Y and protein Z will have tryptophan and X will have tyrosine. So either statement 2 or statement 4 is correct. Now, the emission spectrum of tryptophan is sensitive to solvent polarity. If tryptophan residue is buried in a hydrophobic environment within a protein, the emission may be blue shifted and is maximum near the lower limit. Okay, so if it is buried inside, right? So it will show an emission spectrum near the lower limit. Lower limit means near 300, and if it is on the surface, it shows a shift towards the higher limit. Okay, it shows a shift towards the higher limit. So if the tryptophan is not buried inside, it is on the surface, it will show an emission spectrum near 350. Okay, and if it is buried inside, it will show an emission spectrum near 300. So you can see here, so protein X is the tyrosine, protein Y has got tryptophan buried inside, and protein Z has tryptophan on the surface. So option 4 is correct. Okay, already option 4 is written over here. So option 4 is the correct answer. Okay, now now let us let us go through another question. So I suppose that is clear. That is just a basic fact based question. Now here also we have a basic fact based question only. So absorption spectra of L-tyrosine in acidic and basic media was estimated and plotted on a graph as depicted below. So you have L-tyrosine. Okay, L-tyrosine means what? Levorotatory, dextrorotatory. What is L-tyrosine? Left-handed tyrosine or right-handed tyrosine? Okay, so what we have, sorry, there is some issue, but still I can continue. Yeah, I suppose you can see my slide. I'll just continue. Okay, so absorption spectra of L-tyrosine in acidic and basic medium is depicted over here. So following interpretations were made. Changes in the pH from acidic to basic results in shift in the lowest energy absorption maximum and decrease in the molar absorptivity. It's very difficult to actually understand, but still I'll make you understand that. Now, shifting of the absorption band to longer wavelengths signifies a shift to lower energy and also known as redshift. Now, this is something which you should be aware of, that a longer wavelength is always associated with lower energy. Longer wavelength means lower energy, right? Always. Why? Because you know, E is equal to h nu. There is a formula, h nu. Nu is nothing but the frequency, and if you convert that into and nu is equal to, and there is another formula which is nu is equal to c lambda, or yeah, nu is equal to C lambda, something like that. So basically, no, 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 no. C is equal to nu into lambda. So basically, nu and lambda, they are inversely related. So E is always, energy is always inversely related with your lambda. Okay, E is always inversely related with your lambda. So if anything has got higher energy, they will have lower wavelength, and if it has got higher wavelength, then the energy will be low, right? Okay, so this is correct. Shifting of the absorption band to longer wavelength signifies a shift to lower energy, also known as redshift. And shifting of the absorption band to a shorter wavelength signifies a shift to higher energy. Shorter wavelength, certainly higher energy. So that is known as blue shift. So B and C is correct. Now let us read through the D option also. So wavelength shift is always accompanied by change in intensity of the absorption band. No, it is not. Okay, so since B and C are correct, let us find out. Yes, option 3. I suppose option 3 is correct. Yes, B and C is correct. Okay, so this is once again from the common understanding, from the normal understanding, that a longer wavelength always signifies to lower energy, and a shorter wavelength always signifies to higher energy. They are inversely related. Okay, they are inversely related. Fine, students. Now I'll go to another question. So here we have it's a very simple question. A multimeric protein, when run on a SDS gel, showed two bands at 20 kilodalton and 440 kilodalton. However, when the protein was run on a native gel, it showed a single band at 120 kilodalton. The native form of the protein would be, since 120 120 is the actual mole weight of the protein, 120, and you are getting two bands. Okay, one of 20 and another one of 40, so together they will make 60. Okay, so another 40, 20 and another 40, that will also make 60. So actually the protein is made up of 1, 2 and 3, 4, right? 4 polypeptides, and hence hetero it will be a heterodimer. It should be a heterodimer. It should not be a heterotetramer. I'll recheck this question. It should be a heterodimer. It will not be a heterotrimer. It will be a heterodimer. Okay. Now the next one is, uh, before this one, I'll solve this one for you. Or one more is there. One second. No, let's solve this one. Okay. So this one, the answer should be heterodimer. Okay. Fine, students. Yes, it should be heterodimer. Heterodimer, not heterotetramer. If anybody is saying heterotetramer, please explain. Please explain. It should be heterodimer. Okay. Now here we have an now we'll come to the thermodynamics questions, which is there, and here also we will yes, Shivani. So here we will use some formulas, and I want to introduce that formula to you. Since time is very less, I will actually try and uh, tell you how it is solved. So let us read the question first, and then I will show you the calculation. I have already done the calculation for you. So here what we have, DNA melting temperature, that is Tm, was found to be at 47 degrees Celsius, and enthalpy measured at Tm was 0.032 kilojoule. The entropy change would be, so here the value of melting temperature, that means temperature is given 47 degrees Celsius, the enthalpy, that is your H, is given 0.032, and they're asking what is the S, entropy change. Okay, delta S. Okay, so there is a formula which we use, and that formula is this one. Okay, so here we have the formula. And what is the formula? Delta S is equal to delta H by Tm. Now Tm is in degrees Celsius, so we'll convert that in Kelvin, and it will be 320 Kelvin. So delta S will be equal to delta H divided by 320. That will be equivalent to this one. So this is your B is the answer. So simply we are using this formula. There are 3 to 4 different formulas. I will introduce you here itself. But for this one, the formula is delta S is equal to changes in enthalpy divided by temperature. Okay, changes in enthalpy divided by, changes in entropy is equal to changes in enthalpy divided by temperature. Okay, so simply you put this formula and you will get an answer. Now, the next question, let us do this. This one is also from CSIR paper. The standard free energy delta G naught per mole for the reaction A to B at 30 degrees Celsius in an open system is minus 1000 calories per mole. What is the approximate free energy change, that is delta G, when the concentration of A and B are 100 micromolar and 100 millimolar respectively? So here what we have to do, we have to first write what is given in the question. So what is given? The temperature is given, that is 30 degrees Celsius. Okay. The delta G prime naught is given, the standard free energy change is given, and the concentration of A and B is given. And what we have to find out? We have to find out delta G. So basically we have to find out delta G, and delta G is equal to nothing but delta G prime naught plus um, that is RT natural log of K equilibrium prime. Okay. Now K equilibrium prime is equal to concentration of product divided by concentration of reactant. Okay, so just you put all these values and you will get an answer. So delta G prime naught is nothing but 1 minus 1000 kilocalorie plus, what is the value of R in calorie? 1.99. Okay, into temperature, that is 30 degrees Celsius to 273 plus 30, that is, what is 273 plus 30? 303. So into 303 into log of, and since it is natural log, so we have to put 2.303 over here also. I'll show you the calculation. Yeah, see the calculation, it's very clear. I've already written. So delta G is equal to delta G prime naught plus RT natural log of product by reactant. Standard free energy is given, R value is 1.99 or 1.98, temperature is 303. Now substitute all these values. Now since it is natural log, so you have to multiply this with 2.303. You can see over here. So delta G prime naught plus 1.99 plus into 303 into 2.303 into log of product concentration divided by the reactant concentration. So you will get this as the answer, 3155 calorie per mole. Okay. Okay, students. Okay, students. So these were some of the questions which had already come in exam, and I tried to discuss all those questions with the concept, so that if anything from this concept comes in exam, you can easily touch those questions and answer them. Just you have to do what? You have to inculcate the reading ability. You have to know how to read those questions, right? And most of the questions, they are directly based on the concept. Okay, and little bit of logical reasoning you need to have. Okay, I hope you enjoyed the session, and if you have any query, you can always come back to me on crisp chat. I will be happy to solve your queries, and all the best for your CSR exam. Thank you. Good night.